Welcome, everyone. I am Pei Ping, together with Dr. Carol Watson from the Skills and Workforce Development Core of the NIH Bridge to AI Center. We present you on the fundamental concepts to AI and ML education module, led by Professor Wei Wang, Computer Science, UCLA School of Engineering. Welcome, Wei.
Thank you, Pei. And today, we are honored to have Professor Jingyi Li to give a lecture on statistical learning. Professor Li is a professor of statistics, biostatistics, human genetics, and computational medicine at UCLA. She's currently also a Helen Pullman Fellow at Harvard. She received her PhD from UC Berkeley, and her research focuses on developing statistical and computational methods to answer important questions in the biomedical field and to extract key information from genetics, genomics, and health-related data. She has been recognized with numerous awards, including a Sloan Fellowship and NSF Career Award. In this lecture, she will introduce key concepts in statistical learning and in particularly how it's related and distinct from traditional machine learning. Without further delaying, I'm going to hand over to Professor Li to start her lecture.
Thank you, Professor Wang and Professor Ping for the nice introduction. It's my pleasure and honor to present this lecture. So it's about the clarification of two fundamental formulations widely used in data analysis: statistical hypothesis testing and machine learning binary classification. And I will talk about each and clarify their differences. And the reason why I think we need clarification in real data analysis between the two paradigms or frameworks is because of this application. So this is a paper published in Cell in 2013. And this paper uses computational methods to predict whether an unknown cancer driver genes or non-cancer driver gene, like a gene A marked as unknown in gray, is a cancer driver or not. So in our knowledge, we only know a small proportion of genes to be cancer driver or for sure to be non-cancer driver genes. And here by cancer driver, we mean pan-cancer. It's not specific to one type of cancer. And in this paper, this data was manually curated from public data. And here the features for every gene are some genomic features in DNA, including the mutation frequency. So the goal is to use those features and to use our knowledge of cancer driver genes and non-cancer driver genes to predict whether an unknown gene is likely cancer driver. That's the goal. And the approach taken by this paper is summarized in the bottom diagram. So specifically, the researchers wanted to obtain a p-value for every unknown gene. The reason is because p-values are widely used in computational biology. So I will talk about this later, but basically p-value is a concept used in statistical testing. And in their formulation, they want to do one test per unknown gene. And because we have many unknown genes, this is called a multiple testing problem. So how do they do the test for every gene? Specifically, let's say we have this gene A to be examined, and we look at each feature one by one. Say we look at feature 4, and we use the non-cancer driver genes values in this feature 4 and consider these values as null values. We want to compare this gene A's value to the set of null values so we can assign a p-value to gene A for this feature 4. How do we do it? We know that p-value is defined as the probability that we observe this value or values more extreme than this value under the null. So then we need to define what is considered more extreme. Is it bigger than or smaller than? That direction is determined by cancer driver genes values. So you can see that in this formulation, cancer driver genes values were not used directly, but they were only used to direct the direction for p-value calculation. So suppose we know the direction to be larger than, then we can get this p-value for gene A for feature 4. But we want to just have one p-value for each unknown gene A. So this approach can be applied to every feature. So we would obtain d p-values for gene A. And then the authors used Fisher's method to combine the d p-values into one p-value for gene A, called PA. So this approach can be applied to all the genes. And let's say we have every gene with a p-value and have many p-values. The last step is to set a threshold to satisfy some criterion. And the most famous criterion is called a false discovery rate, FDR. To control the FDR by threshold in the p-values, the procedure is called Benjamini-Hochberg procedure. So it will give us a p-value threshold. And then for all the genes with p-values under this threshold would be predicted as cancer driver genes. This is the framework in the Cell paper, and it takes the multiple testing framework. And when I know this formulation, my first question is, Fisher's method seems to be not the appropriate one because it has an internal assumption that every p-value or all the p-values are mutually independent. But in this case, since the D features were manually curated, it's very likely, or we can actually see it from this matrix, that some features are highly correlated. So that independence assumption is problematic. And furthermore, I think the fact that the cancer driver genes values in every feature are not used directly, that may be another concern because we didn't fully use the information. And finally, the features dependency, right? Because we want to use D features jointly to make the decision, is not naturally captured in this approach because we examine one feature at a time. So all these issues motivated me to think, can we use a simpler formulation, which is binary classification? Because binary classification can naturally handle P features as the input. And we are in the scenario that we have the two classes, cancer driver genes and non-cancer driver genes. So this is the scenario. We have two classes and every gene has a label, right? Some are cancer driver, some are non-cancer driver. And for the majority of genes, we don't know their labels. So this is a prediction problem, or binary classification belongs to supervised learning. And supervised learning is one type of learning, right? We have supervised learning, unsupervised learning, as you have heard from Professor Wang's lecture before. So using this binary classification formulation, the cancer driver genes data in those D features and the non-cancer driver genes data in those D features would form a training data set, from which we can use a classification algorithm, be it support vector machine, a tree classifier, or its more complicated version, random forest, or the neural network. We can apply these algorithms here, and each algorithm after training would give us one classifier. So using this classifier, we can input each unknown gene, say gene A, its p-dimensional feature vector as an observation into this classifier. And then we can predict whether the gene is more likely to belong to cancer driver class or non-cancer driver class. So then we will still have a probabilistic output if the classifier itself is probabilistic. So to me, this formulation naturally accounts for the dependency among the D features. So we don't need to worry about that independence assumption in the Fisher's method. So motivated by this, and also I have to give credits to my collaborator at UC Irvine, Dr. Wei Li, who is a computational biologist who studied cancer. And so he brought this problem to my attention because his project wanted to add epigenetic features to genetic features in the cell paper to achieve better prediction of cancer driver genes. But when I heard about their project and learned about this multiple testing formulation in the cell paper, I had the questions I just said. So I just proposed to modify the framework to be binary classification. And that's how we co-published this paper in Science Advances to implement this binary classification formulation. So this example gave me the motivation to say, yeah, it seems that in real data problems, the distinction between using multiple testing in which we have every gene with the test versus binary classification in which we input every gene into a trained classifier, the distinctions are not as apparent as we thought in textbooks. So how do we formulate it? And this gave me an opportunity to write this perspective article to clarify their distinctions and to provide some guidelines. And this is co-authored with my collaborator, who is a statistician in the business school at USC, University of Southern California, Dr. Xin Tong. So we both teach classes. And from his teaching experience and my teaching experience, it's interesting that students learned hypothesis testing in the statistics class, like introduction to statistics class, where people learn about the null hypothesis, alternative hypothesis, p-value calculation. But for binary classification, students learn it from a machine learning class. So usually the two topics will not be covered by the same class or same textbook. So that makes the students, when they learn them each, they don't need to do the comparison, right? They will think, oh, this is that, this is this. It seems to be different. But when you face a real data problem, then you need to make the decision, which formulation should I adopt? So to help with this decision, we wrote this perspective article, and that will be the focus of my talk today. So now let me change the example to be something more intuitive, disease diagnosis. So here I label the rows as patients and label the columns as genes, or in general features. So basically, if we have patients labeled with not without the disease, no, or yes, with the disease, and we want to predict if a new patient has the disease or not. That's the disease diagnosis setting. I think in this case, it is more straightforward to think, this is a classification problem. Okay, so we want to train a classifier based on the training data of disease patients and undiseased patients, and so we can predict whether a new patient is a disease. So this is what we call a learning problem. So I'm here using the language as used in machine learning to call patients as instances, genes as features. And in statistics, people may use other terms like instances are referred to as observations. And also in machine learning, some people may refer to instances as samples. So these terms were used exchangeably. And features, some people may call them variables, even covariates. So we also have more than one term for the features. But to make it simple, I will stick with instances and features for the rest of my talk. So how do we differentiate the two? My perspective is that features are some quantities you want to measure, and they are permanent. So you, like, for example, genes are permanent. Our human has more than 20K protein coding genes, and these genes won't change if we measure different people, right? So we always have those genes existing. But patients, we consider them as a random sample from our human population. So in different studies, we have different patients. So instances can be regarded as random observations from a population, while features should be regarded as fixed quantities we want to measure. So given this perspective, binary classification is trying to answer questions for instances. For each instance, we want to make a binary decision. So it's called a learning problem. And I will revisit this concept again later. But testing, hypothesis testing, the question is not about a particular instance like a patient one, but it's about the whole population. So in this case, if we're interested in this column gene A, whether it is a biomarker, then the question is, does this gene A have very different expression levels between the disease patients and undiseased patients? So this question, we're trying to answer it for the whole human population, not just for the particular set of patients we have data for. So in statistics, this type of question is called inference, because we are interested in making inference about the population, not just about this particular sample. So you can see in this case, to answer this inference question, we will take gene A's value in disease and undiseased patients, and to compare these two sets of values to run a statistical test and to decide whether we want to reject the null hypothesis that gene A is not a biomarker or not. So this more intuitive example may let you see a better distinction between the two frameworks. So then I'll give more specifics. To clarify the distinctions, I summarized the 4 concepts. So to help us understanding, so what's the difference? What's different between testing and classification? So the 4 concepts are binary classification, binary answer, decision rule, binary decision. So binary question is the nature of our question, right? So it's the nature of the thing we want to do analysis for. So in the hypothesis testing framework, the binary question is, do we think the null hypothesis is true or false? In binary classification, is that do we think the instance has labeled 1 or 0? So you can see that this is instance specific, and this is about the hypothesis. And hypothesis is about the whole population. So I want to clarify that the binary question in hypothesis testing is actually unanswerable. The reason is because our data is only a sample from the population. If we don't observe the whole population, like everyone in the world, we can only say how much we trust the null hypothesis being true or false, but we can never know whether it's really true or false, because there's always discrepancy between our sample, no matter how large it is, and the population. So that's the way statistics thinks. And for the binary answer to the question, so basically, in testing, 0 means we think the null hypothesis is true, 1 means the null hypothesis is false, but again, the 2 answers are not observable because we don't observe the whole population. In binary classification, the instance has labeled 0 or 1, and these may be achievable because in our training data, we do have those labels for instances. Okay, the most important thing is decision rule. What is the rule in hypothesis testing? It is a statistical test that inputs data and outputs a p-value, which is compared against a user-specified significance level alpha. In other words, we compare p-value to alpha. If p-value is smaller or equal than alpha, we reject the null hypothesis. So in this case, it's our binary decision. 0 means we do not reject the null. 1 means we reject the null. But we have to note that here the decisions may not be the correct answers. So that's why we have ways to measure the errors we make in hypothesis testing. In binary classification, the decision rule will be a trained classifier on training data that inputs an unlabeled instance feature values and outputs a predicted label for that instance. So in this case, the binary decision output by the rule will be a label as 0 or 1. So even though both make binary decisions, but the key difference is about the nature of the question they try to answer and the decision rule. So here I'm going to show one example to make the point more explicit that in the decision rule of hypothesis testing, we don't need training data. And this is different from binary classification. So I'm going to talk about the most famous students 2 sample t-test in this case. Suppose that we have 2 samples of sizes 10 and 12 from 2 normal distributions. And we're interested in whether the 2 normal distributions have the same mean. Then the null hypothesis is that the 2 normal distributions have the same mean, and the alternative hypothesis is the opposite. Here, the test statistic, which is the 2 sample t statistic, follows the t distribution with 20 degrees of freedom, under the null hypothesis. So this is a well-defined distribution. We have a table to look up in the old days, and now we can easily do the computation in R or Python. Then given a significance level alpha between 0 and 1, many people set it to 0.05 by convention. We would reject the null hypothesis if the t statistic has an absolute value greater than or equal to this particular value. So we can ignore this notation. Basically, it's a threshold determined by alpha and by this t distribution, which is this quantile of the T20 distribution. So for example, using the convention alpha equals 0.05, this threshold is about 2.08. So if our t statistic has an absolute value greater than this 2.08, we will reject the null hypothesis. So you can see that up to here, we didn't have any training data. This whole procedure, decision rule, the threshold were based on theory. So you can see that this decision rule is equivalent to that the p-value is less than or equal to alpha. Note that this decision rule does not depend on any observed t statistic value calculated from a data set, right? The rule is set by this threshold and the formula of the t statistic. However, a decision itself, not a decision rule, but the decision requires an observed t statistic value. For example, if the t statistic value has an observed value of 3, then we would reject the null hypothesis at alpha equals 0.05. So even though the rule is set, not depending on training data, but you need to input your data to use the rule to make a decision. So that's what we have in hypothesis testing. Okay, but we know that for binary classification, just go back a little bit, you have to rely on training data because this classifies your rule. Otherwise, you wouldn't have this rule. So training data is indispensable in binary classification. And the second table in my paper did a side-by-side comparison of hypothesis testing and binary classification from multiple aspects. So the first one is whether the binary answers have symmetry. So in hypothesis testing, the default is the null hypothesis. So the idea is that you wouldn't reject the null hypothesis unless you have a strong enough evidence from your data. Otherwise, you stay with the null. But I have to clarify that staying with the null only means you don't have strong evidence to reject it. It doesn't mean the null is necessarily true. So this is the asymmetric, a very conservative perspective taken by hypothesis testing. But binary classification, in most cases, it's symmetric. You want to predict the two classes, class 1 and class 0, equally well. That can be reflected in your loss function. But more recent, there are developments to allow that two classes are treated asymmetrically. So like one class can receive a higher priority. So you want to make sure it's predicted accurately, and you're willing to sacrifice the other class for this accuracy for the more important class. So that can be changed by the loss function. But this is just the default. That is, hypothesis testing is asymmetric, binary classification mostly symmetric. And the second aspect I think is very interesting is that how many instances do you need to make one decision given a decision rule? So in hypothesis testing, you want the sample size to be the larger, the better, right? Going back to this biomarker example, so you want these two groups of patients who are diseased or healthy, right? You want these two groups of patients to have a lot of patients, right? So then you can have a good power in this test. So the larger the sample size or the more instances you have to make a decision, the better. But in binary classification, when you want to label each instance, you only have one instance to label. So you input one instance, as in this cartoon. Here you input one instance into the classifier, and the instance receives a label. And the third aspect is the availability of binary answers. As I said, in hypothesis testing, we don't know the answer because that's the ground truth about the population. We don't know it. But in binary classification, yes, we know the answers in the training data. They are labeled. The fourth aspect is the evaluation criteria. So in hypothesis testing, it is the power, because we already fixed the significance level, and the larger the sample size, the better the power we have. Or given the same sample size, some test statistic may be more powerful than others. So power is the criteria. And in binary classification, the criteria is prediction accuracy. The prediction accuracy is how well do your predicted labels agree with the actual labels. So you often do this by using cross-validation on your training data. You leave out some data, some instances with labels, as if you don't know the labels. You apply the trained classifier on the remaining training data, and then you compare the predicted labels to the actual labels of the training data you use for validation. So that's the idea of cross-validation, and use prediction accuracy as the criteria. So the last aspect is something often ignored by people, but I think it's very interesting to mention, is that what if you have the largest possible number of instances? That means, what if I observe the whole population, everyone in the world? Then in hypothesis testing, as long as the test is valid, the power should be equal to 1. Why is that? The reason is because, let's say we imagine we observe everyone in the world, then we would know for sure whether this gene A is differentially expressed between diseased people and normal people, right? So we can know this answer for sure. Then we can say, yeah, if gene A is indeed different, we can for sure tell it. That means power equal to 1. But the binary classification accuracy may not be equal to 1, even though you observe everyone in the world. The reason is that, coming back here, if you have these training data, right, these are the disease patients and the healthy patients. If you observe everyone in the world, but some patients may be hardly distinguishable, right? In terms of the D features, you can hardly tell whether they are disease or healthy because they may be similar, alike. So the two groups may have some overlap. So in this case, even though you have the best data possible, it's still very difficult to, it's still impossible to tell for sure whether a patient near the boundary is diseased or undiseased. So that's why binary classification wouldn't have perfect prediction accuracy, even though you have the largest possible data. So the second box is trying to clarify or say what I just said more specifically. So I'll just read it. A general principle in data science is if a sample is unbiasedly drawn from a population, the larger the sample size, the more information the sample contains about the population. This large sample principle holds for both hypothesis testing and binary classification. For example, data from a larger number of representative patients will lead to better decision rules for both biomarker detection and disease diagnosis. However, between the two strategies, there is an interesting but often neglected distinction. From a population with finite instances, such as the human population, the largest possible sample, which is equivalent to the whole population, would make a valid statistical test achieve a perfect power given any significance level alpha, while the largest possible training dataset might not lead to a classification rule with perfect prediction accuracy. While this distinction is fundamentally rooted in mathematics, an intuitive understanding can be obtained from our biomarker detection and disease diagnosis examples. Imagine that we have measured everyone in the world. If a gene is indeed a disease biomarker, we can for sure see a difference in this gene's expression between all the people carrying this disease and the rest of the population, achieving the perfect power. On the other hand, disease patients and undiseased individuals may not be perfectly separated by diagnostic measurements. That is, two patients may have similar symptoms and lab test results, but one patient is diseased and the other is not. When this happens, even if we have training data from all but one person in the world, we still cannot be 100% sure whether this left out individual has the disease just based on his or her diagnostic measurements. So in the following, I'll talk about the specific guidelines for making decisions in real practices. So let's say we still use this example. Rows are patients, instances, columns are genes, features. And so how do we decide on instances and features, right? I already gave the answer just now, but let's say if we only have this matrix, and rows could be instances or columns could be instances, because we can always do a transpose on the matrix. So I would say mostly for instances, they are repeated measurements in the data collection process. If you repeat your experiments for multiple times, then you can regard every experiment's output as an instance. Or the instances can be a random sample used in statistics, or random samples, the plural form, used in machine learning from a population. So in this case, the patients belong to the second category. So that's why we will say they are instances. And instances and features. That's the guideline 1. Try to decide rows and columns, what each one is. Guideline 2, list the binary decisions to be made. So in most cases, if the questions are concerning instances, you can use a learning strategy. For example, binary classification belongs to supervised learning. Or if the questions are feature specific, then it's an inference problem, and hypothesis testing is an inference procedure. The third guideline is to assess the availability of binary answers in data. Do we know the answers for some data or not? So in most cases, if you do have binary answers available, think about binary classification, because these can be your training data. That if you don't know any answers at all, think about hypothesis testing, because your questions may be about population, so you would never have the answers because you don't know everything. Guideline 4, count the number of instances for making each binary decision. So in most cases, if you're making one decision per instance, think about binary classification or learning problem. If you need several instances per decision, and the more instances, the better, think about hypothesis testing, because you want to use a large enough sample to infer about the population. The larger the sample size, the better. Guideline 5, evaluate the nature of binary questions. So this is the most fundamental one. If you know this one, then probably you won't need the previous guidelines because they are more practical, and this is more fundamental. So basically, the question, if the question is regarding a particular instance, think about learning problem binary classification. Or if the question is regarding an unseen population, think about hypothesis testing. So these are the most important messages from this perspective article I wrote, and you can check it out for the more detailed descriptions, and I'll be very happy to answer any questions you may have. So finally, I want to thank my co-author, Dr. Xin Hong, for that perspective article. I also want to thank my colleagues in academia and my students for their very insightful comments on this manuscript before it was published. Especially, we posted this manuscript to the archive, and we got an email from Dr. Thomas Berger, whom I didn't know before. He worked in France, and he works on proteomics research. He said that in the proteomics field, he saw many people confused between hypothesis testing and binary classification. So that's why he found our article relevant and useful for the computational researchers in his field. So I will stop here so we have more time for questions, and I'm very willing to answer any questions or your comments.
Thank you, Jessica. So now we have time for questions. Please post your questions on Q&A panel.
There's one question about the recording. Yeah.
Yes. So I think Dr. Pei Ping is going to answer that question.
Yeah.
Hi, we have from Johanna Alvin, have a question regarding the lectures for this specific one and the other lectures that Skills and Workforce Development Core has organized. We're actually offering the Bridge to AI community as well as the broad scientific community a whole library of video recordings we currently have constructed on a live internet platform. It is currently under approval by both University of California at Los Angeles guidelines, as well as harmonization with the entire Bridge to AI program website and platforms. And very soon it will be launched and released to the public. And we're happy to share a link with you if you have an urgency, would like to have it sooner.
Thank you. And I see that Jing Yuan asked for the link to the article. I typed it to the answer, and I don't know if all the participants can see it.
Yeah, I think, well, because I think, yeah, Jing Yuan said thanks.
Yeah.
I think she's able to see it.
Yeah, and I'm also typing it to the chat to everyone, so everyone on the Zoom should be able to see it. Okay.
So we have another question coming in.
Yeah.
Would you please explain how to correct the multiple comparisons problem, especially? Yeah. I think it's not finished. I'm wondering if there is an alternative to buffer running correction.
Okay, good question. Actually, if I go back to the beginning of my talk, here I said in the cell paper, it uses the Benjamini-Hochberg procedure. Okay, so it's another way in addition to or alternative to buffer running correction. And here, why we have two different correction procedures? The reason is because we have different criteria defined for controlling the error of all tests together. So if it's easier, I can type in the chat box. That is, the Bonferroni correction aims to, the criteria is to control the so-called family-wise error rate, short as many people wrote it as FWER. And this is defined as the probability that at least one test decision is wrong. So it's a very conservative correction procedure because we want to make sure that the probability that we make one mistake is very small, is controlled. And you can imagine that this probability family-wise error rate would increase as your test number increases, right? The more tests you have, the more difficult it is to control the family-wise error rate. And so Bonferroni correction is very conservative because you need to divide each p-value by the number of tests before you compare the corrected p-value to alpha. So the more tests you have, the more correction you're doing, and then the fewer tests you would reject. So that's why using Bonferroni correction, when the test number is big, you may not reject anything. So scientifically, you don't make any discoveries. That's why people don't like it. But later, the Benjamini-Hochberg procedure correction, this one aims to control the false discovery rate, short as FDR. And the false discovery rate is defined differently. It's defined as the expectation of expectation of the ratio. The ratio is that the number of false discoveries over the number of discoveries. So let's say that given a threshold, you reject those tests under the threshold with corrected p-values under the threshold, and then you would say, if I know the ground truth, among my discoveries, what proportion is false? That's the ratio. But the interesting thing is that even though this ratio is never observable, because we don't know the ground truth, but somehow this Benjamini-Hochberg can control the expectation of this ratio. By expectation, we mean what is the average if you observe many possible data sets like your data set from the same population. So we are doing this thought experiment. We imagine possible potential data sets. And this expectation is the ratio calculated across all the data sets. So the magic of statistics is that even though on your data set, the real ratio, the particular ratio, you don't know the value, but you can control the expectation. And this magic lies in the fact that for the p-values from the null hypothesis, if the null hypothesis hold, the p-values should be uniform between 0 and 1. And that's the theoretical foundation of this Benjamini-Hochberg correction procedure. So back to Dr. Kim's question, I want to say the key difference is that these two correction procedures both want to control something by taking all the tests together. But Bonferroni corrects for something or controls something more stringent, family-wise error rate, and Benjamini-Hochberg controls something less stringent, FDR. So using the FDR control criterion, we can find more discoveries. That's why FDR has been very popular nowadays.
Thank you, Jessica.
Yeah, hopefully I answered this question.
Next question is, does it ever happen that you achieve high binary classification result, but non-significant statistical results? If so, how should you interpret the results?
Let me clarify this question. First, what do you mean by high binary classification result? Because I guess you might mean high accuracy in binary classification. I think my point for this lecture is that the two frameworks are not comparable. We shouldn't compare them in terms of performance because they answer different questions. Maybe you were trying to ask this example, right? The one I questioned about in the beginning, because the original paper used this, what I think as inappropriate multiple testing formulation. But in my change, I used the binary classification formulation to redo the problem. And I can tell you that empirically, even though we treat this multiple testing idea as a way to do this problem, and we use the same evaluation criteria about accuracy, this binary classification gives us better accuracy. So empirically, we should also use binary classification. But maybe back to your question, Dr. Chen's question, once you say non-significant statistical results, I'm guessing you're saying the p-values are not significant, I guess. But in this case, my point is that if you think the problem should be a binary classification problem, that is, your decisions are made for instances, not features, then you shouldn't apply statistical testing to instances. That doesn't make sense to me. So that's why my take-home message is that they shouldn't be compared because they are different in nature.
Thank you. The next question. Statistical inference requires fitting a proper sampling distribution. If we don't have the prior knowledge regarding the sampling distribution of a data set, how do you find out the appropriate distribution that can be used for testing?
That's a very good question. I want to clarify that by fitting a proper sampling distribution, I don't know if the Dr. Wang, who asked this question, thought about parametric distribution like Gaussian distribution, Poisson distribution. And I want to say that we don't have to. So that's why in statistics, there is an area called non-parametric statistics. And the most famous non-parametric test is the Wilcoxon rank sum test. So using the Wilcoxon rank sum test, we don't need to assume that the data follows a normal Gaussian distribution or any other parametric distribution. We just need to have some independence assumption, which is about the sampling. Do you believe that your instances were independently sampled from a population? And that's the question our data analysis need to think about, right? When we do statistical formulation, independence is the first thing we have to keep in mind. So, but I have to say, compared to assuming the data follows a Gaussian distribution, independence is something much easier to check using our knowledge about the data collection process. And yeah, so basically we have this tool set for doing non-parametric tests. And if your sample size is large enough, like in this problem, right, to compare the diseased people to the undiseased people for this gene A, if we have a large sample size, by large, I mean more than 20 individuals in each group, we could apply Wilcox or Wrexham test. And we are not assuming the data to follow a specific form of distribution. I hope that clarifies.
Thank you.
Yeah.
Our next question is from anonymous attendees. I have a general question. What's your opinion in dealing with data imbalance in ML? Do you consider data balance for statistical testing?
Very insightful question. Yes, data imbalance is an issue in both cases. So in machine learning, people often think data imbalance will make the training more difficult, or your trained classifier may have worse prediction accuracy. I have to say, maybe Dr. Wang can help me, but based on my knowledge, I don't think people have a consensus about how do we improve the data imbalance, right? Should we do resampling, like downsample the large class to make the two classes more balanced? Or there are approaches who do oversample the minor class to make data more balanced. But whether such procedure will give you a better classifier than using just the raw data, original data, at least based on my reading from online literature, I don't think there's a clear consensus. Or whether you should make the two classes perfectly balanced, like one-to-one balance. I don't think there's a clear guideline, but people may use cross-validation to check empirically. Do you think rebalancing will give you a better cross-validation prediction accuracy? At least that's something you can check. But clearly, I would say if the two classes are severely imbalanced, there would be an issue. So the data wouldn't be as nice as you want it to be. But speaking of that, I would try to say that we have some research along this line, and I can share the link here. And we call our framework the Neyman-Pearson classification. It's for binary classification. And what we aim to achieve is that asymmetrically, basically, that's our perspective. So we will think about a scenario where one class is more important than the other class. So we want to first prioritize the more important class to make sure our prediction for this class is good enough. And then we minimize the other class prediction error. So that's the asymmetric perspective. But a side advantage of Neyman-Pearson classification is that it is robust to class imbalance. So the decision rule found by Neyman-Pearson actually doesn't depend on the class ratio, which I think is a very good advantage if you're really concerned about class imbalance. So that's about the first question. And in the second question, statistical testing, data balance would affect the power. And this is known in genetics, like in GWAS, when people talk about the rare variants detection, GWAS, right? If you have a variant that only has the alternative allele in very small number of people, and for the majority of population, you have just the reference allele, people use the quantity called the minor allele frequency, right, MAF, to measure that thing. So for those rare variants with very small MAF, then you do GWAS, and the p-values wouldn't be small enough for you to call it an association. And the reason is lack of power. So you can think about the power here depends on the sample sizes of both groups. If one group is particularly small, then you will sacrifice the power. But if you do the statistical test properly, you can still control the type 1 error under alpha. So that part you control, but the power is something that will change with the sample size. So hopefully that answered the question.
Thank you. I think those are very thoughtful answers. We don't have any more questions. So if you have any last minute question, please do type into the Q&A box. There's no more question. We would like to thank Dr. Jessica Li again for the wonderful lecture. And we will also thank for all the attendance for this lecture. So we will conclude this lecture, and then we'll see everyone again next week.
Thank you, Dr. Wang, and thank you, Dr. Peng.
Thank you, Professor Li, for such an insightful lecture. I learned very much about myself.
Thank you.
We'll see everyone next time. Thank you, Professor Wang, as well.